Hello everyone, my name is Anthony Siler and I am a junior biology major at West Virginia University, and I had the opportunity to work in Dr. Kolandaivelu's lab this summer on a project involving NMNAT1, which is a protein linked with blinding diseases that is post-translationally lipid modified by protein palmitoylation. Now I'd like to give a little background on what NMNAT1 and NAD are and why they're important to us. So NMNAT1 is a nuclear-specific enzyme that is found throughout the entire body and is responsible for synthesizing NAD+. And as you can see in this diagram to the left, it catalyzes the reaction of NMN to NAD+. And you will notice that NMNAT has three different isoforms. There's NMNAT2, which is localized to the cytoplasm, actually right outside the Golgi, and NMNAT3, which is localized to the mitochondria. And NAD, as seen here, is very important to us. NAD is a critical coenzyme that is found in every single nucleated cell of the body, and it's vital for the regulation of many cellular processes. Most people know that it's responsible for energy production, making ATP. It's also involved in chromosome stability, DNA repair, immune cell signaling, brain health, and enzyme activity. So mutations, specifically in NMNAT1, that catalyzes reaction to NAD has been linked to specific diseases within the retina. And the function of NMNAT1 in the retina and the mechanisms behind retinal degeneration are unknown and not well understood. So we hope to find out more about NMNAT1, the role it plays throughout the body, and specifically in the retina. Now I'd like to give a brief introduction about DHHC. And what it is, it's a protein domain that generally acts as an enzyme to palmitylate proteins. And I'm going to talk about palmitylation here in the next slide. And there are 23 of these DHHC proteins in mammals, and they're also known as palmitoyltransferases. And we know from prior research that some DHHC proteins are known to enhance the palmitylation of other proteins. So this leads us to the question, what is palmitylation? Well, palmitylation is a lipid modification that mediates protein sorting, localization, stability, and subcellular trafficking. So palmitylation can, in essence, change the structure, function, and location within the cell. And here you can see I have a diagram on the left to describe how this palmitylation works. So palmitoyl coenzyme A is basically a lipid donor, and the lipid that it's donating is this palmitic acid, more informally known as a palmitate, and that's a 16-carbon chain. And this specifically occurs on a cysteine protein due to the fact that it has this thiol group as its R group, which differentiates it from the other 20 amino acids. And you see these two enzymes over here. And what's important about palmitylation is that it's a reversible modification. So some parts of the protein will be palmitoylated while other sites will not, because it's constantly in this equilibrium, as you can see, between being palmitoylated and not being palmitoylated. And as you can see, this thiol group interacts with a carboxylic acid, reacts with this acid to form a thioester linkage, as you can see down here. And this is when the protein is now palmitoylated. And we strive to find the function of NMNAT1 by identifying if it is palmitoylated or not and has this thioester linkage, this 16-carbon lipid addition. And our focus is to identify if DHHC enzymes specifically are responsible for this NMNAT1 palmitylation. This leads to our hypothesis, which states that NMNAT1 palmitylation is essential for protein stability and function needed for producing nuclear NAD+, which we previously discussed, and we saw how important NAD+ is and how vital it is for our overall health and well-being. So this now leads us to some research questions. So if NMNAT1 is palmitoylated, which one of the DHHC proteins, because there are 23 mammals, is responsible for this NMNAT1 palmitylation, which enhances it? And if NMNAT1 is palmitoylated, what is its role in protein stability and subcellular localization? So how does this lipid addition change the function of NMNAT1? Where is it now localized to? So before we can do any of this, we need to know if these DHHC enzymes are produced correctly. So how we did this, we transfected HEC293 T cells with DHHC plasmids 1 through 23, and then we collected these cells and we were able to run a Western blot and then probe with antibodies targeting these DHHC proteins to see if they're produced in the correct amount and at the correct molecular weight. So here are the results of that experiment, and right here we're specifically looking at DHHCs 1 through 12. And over here on the left, you can see that this is a marker. This is what we use for reference to identify where these proteins are produced, if they're produced at the right molecular weight, because these are known molecular weights here on the side. And we went through and looked at prior research and where these DHHC proteins are expected to occur, and noted which ones were produced correctly and which ones weren't. And the ones highlighted in red you can see are ones that we determined that did not work. And beta tubulin was used as our control, since we expect to see beta tubulin right at 50 kilodaltons, which we did, and we saw a distinct band across all of the DHHCs. And here are the rest of the results. And these are DHHCs 13 through 23. Again, we determined which ones worked and didn't work. And you can see the very dark bands that we were looking at specifically. We had a little bit of cross-reactivity with beta tubulin. As you can see, there's something in the minus control here and all the way across, which we are not supposed to see. But we're specifically looking at the dark bands where most of the protein occurred at, and to see if these were produced at the correct molecular weights. And again, here is beta tubulin that was used as our control. So now after this step, we are co-transfecting HEC293 T cells with the testable DHHC enzymes along with NMNAT1 and NMNAT2. And NMNAT2 is going to be used here as a control because it is known to be palmitoylated, so we will expect to see a band here, and that's used as a positive control. And some DHHCs are expected to enhance this NMNAT2 palmitylation. And of course, we're going to be running NMNAT1 and DHHC together to see if DHHC will enhance the palmitylation of NMNAT1. Now we're going to assess this palmitylation. We're going to run acyl-resin assisted capture, which is more informally known as Acyl-RAC, and it's a method to evaluate NMNAT1 palmitylation. And then from here, we're going to be running a Western blot to see these proteins. Now here I want to go into a little more detail about the method behind how Acyl-RAC captures our palmitoylated proteins. So remember back when I talked about palmitylation, is that this only occurs on cysteines. And here you can see cysteines have a bunch of free thiol groups. And right here is what we're hoping to isolate by the end, because this is our palmitate. And we use a chemical called MMTS, which blocks all the free thiol groups. So these free thiol groups here on this protein are just going to be blocked with this big chemical, MMTS. So the palmitate is not blocked. So now we use a chemical hydroxylamine, which is going to cleave this thioester bond. So it's going to cleave this bond right here. And this reaction creates a thiol group. So now you have this free thiol group while all the other ones are blocked with MMTS. And then we're going to be using these TPS beads, which are big, heavy beads that you can visibly see, to attach and react with this thiol group because it forms a covalent bond, which is a very strong bond. So now you have this bead here attached, and you're going to be visibly going to be able to be seen in the bottom of the test tube. So now you have this attached to the bead, and the final step is eluting. So the point of eluting is you just want to get this bead off so we can get this back into solution and analyze it via a Western blot. So these are the basic steps involved in this process: blocking the free thiols, cleaving this thioester bond right here, capturing by eluting, which is taking off this bead right here, putting it in the solution, and then analyzing via Western blot. We're still currently working on getting results for this project, so we hope to continue to collect data on NMNAT1 palmitylation and to replicate Acyl-RAC on NMNAT1. And we also hope to inhibit NMNAT1 palmitylation to determine its role in protein stability and subcellular localization. And finally, I'd like to give a special thank you to Dr. Kolandaivelu for giving me this opportunity to work with him this summer, and also thank you to David Sokolov for mentoring me throughout this project.